By standing back here in case you wanted to take notes here, but we're going to go through each of these parts. So we've just talked about glycolysis, which is an anaerobic process it's happening outside the mitochondria. Some cells really basically only do this or do this as a precursor to doing something else. Anyway, basically what's happening is glucose, which is a 6-carbon molecule, is broken into 2 3-carbon pieces. And those pieces are called pyruvate. And the energy yield is 2 ATP and 2 NADH. So remember when we were talking about substrate level phosphorylation, that is the creation of ATP using a phosphate group and enzymes, that is what happened in glycolysis, was substrate level phosphorylation. And later on in the Krebs cycle, we will also gain 2 more ATP in that same way, that is substrate level. So those 4 ATP from this whole process will be generated that way. The rest of the ATP is going to come from the electron transport chain, and we will talk about that separately. All right, so the next stage that we're going to talk about is called pyruvate oxidation, or can be called the preliminary step. Remember that this is happening within the inner membrane space of the mitochondria. So it's just inside the mitochondria, and it's taking those 2 pyruvate and it's going to take off a carbon from each of those, leaving behind a very high energy molecule called acetyl-CoA. I forgot to write those 2 terms there. And anyway, so this is right here. This is 2 pyruvate molecules. Pyruvate, remember, was spelled that way. And these are called acetyl-CoA. And then what's going to happen is I get 2 more molecules of NADH, which are carrying excited electrons. Now, both these NADHs, that is the ones that are now currently in the cytoplasm, and the ones that are currently in the, in this case, the intermembrane space, both of them will have to be transported into the matrix in order to start the electron transport chain, which starts in the matrix and goes across the inner membrane, and in the process, and we'll talk about that later. All right, so the Krebs cycle is the next part to this, and I'm going to take these acetyl-CoA, that is the 2 acetyl-CoA that were made there, 1 from each molecule, and I'm going to go through a cycle where I'm breaking up the carbons that are in the acetyl-CoA and making carbon dioxide, but I get a huge energy yield. So this is where most of the energy is taken off that original glucose molecule. Up to now, it's been small amounts of energy, and in this case, the acetyl-CoA, which is a very high energy molecule, will do this process. Okay, so this is happening within the matrix. So that acetyl-CoA from this process, which is in the intermembrane space, has to be transported. Actually, it'll facilitate its flow there because the acetyl-CoA will be being used, and there are proteins that allow it to go across. All right. So anyway, so what's going to happen? Let's look at the process a little bit closer. So this is kind of the final view. It's in your textbook of the process of the Krebs cycle, also called the citric acid cycle. So let's go through it little bits at a time. It's called the citric acid cycle because the first stable compound in the cycle is called citrate, or otherwise citrate or citric acid are the same thing. All right, so in this process, the molecule oxaloacetate is going to pick up the acetyl-CoA. So I call this the pickup molecule. Notice that it only has 4 carbons. And when it picks it up, it's going to make a 6 carbon. In other words, these 2 things are added together to make this 6-carbon molecule called citrate or citric acid. Now, there are various changes that happen to the molecules, and again, I don't really want you to memorize each one of these things, but I want you to have looked at this process enough where you're not confused by the names that are in here. In other words, like they're not Greek to you. So make sure you look at them. You don't have to memorize them, but you should at least know that oxaloacetate is the pickup molecule, and citric acid or citrate is the first stable compound. Then some things, this basically got changed around. So if you look at that, you can kind of look at the rearrangement of the molecule. I'm going to take off some oxygen. I'm going to take off some excited electrons here, so I get some excited electrons from this particular part. And I'm going to, the molecule is changed a little bit again. I take some more excited electrons off, and also oxygen, or I'm sorry, carbon dioxide. It's not, these are carbon dioxide, sorry. I said oxygen, they're not, they're carbon dioxide. I was wondering why that was happening. Anyway, carbon dioxide. So in this case right now, these two carbon dioxides have broken that acetyl-CoA basically apart, and I've gotten excited electrons from there. Now what I end up with down here at the bottom is I end up with then a molecule where I can then make the ATP from this change to this change. In other words, I can use that energy to phosphorylate and make this ATP. Now this is substrate level. It uses this is another cyclic nucleotide, GTP, as a kind of an interim for the phosphate group that it's adding on there. Now, this molecule then, this whole part over here is just to regenerate this oxaloacetate, which is the pickup molecule. But notice I also get some more excited electrons. I get FADH2, which is carrying excited electrons, and I get another NADH, which is also carrying excited electrons. So basically the cycle is kind of changing forms of the molecule. It picks up the original high energy compounds, it takes off the excited electrons and breaks apart the carbons from that, and then I will use the leftover energy to make ATP and glean some more excited electrons out of the process. So the whole process, so this was one acetyl-CoA, I end up with 1, 2, 3 NADHs, and for 2 acetyl-CoA, that would make 6. I end up with 1 FADH2, and with 2 acetyl-CoAs, that makes 2, and I end up with 1 ATP, so 2 acetyl-CoAs, that makes 2. So this is the energy yield for 2 acetyl-CoAs. Okay. Now what's going to happen, and we are going to talk about this a little bit more, and that is that I'm going to take these excited electrons on NADH and FADH2, and I'm going to use those electrons in the electron transport chain to make ATP.